punish him. Kill the guy now. You have the compound. Beat him. What's wrong with you?
What? What is this?
Good day, sir.
You are watching Jackie Pan in my house.
No, it's King Kong, sir.
King Kong. You've never heard of ACTV before in your life? ACTV is very educative, informative, and there are football sport there. There is entertainment. There are a lot of things you can even attend lectures here. All you know is watch all those nonsense artists and do nonsense, or you watch nonsense on TV. Last night you almost came in bed with your fist.
It's not me, sir.
And you wake up every morning, the bed will be so rough, like you'll be fighting kung fu in your dream. That's the reason why you have F's in your courses, like especially made for you.
I have you last semester.
We if pounds on you. Very stupid though. Yeah, change that section to ACTV right now. And oh God, the baby wants to have the first born. Stupid boy.
Good day, everyone. Welcome to both 203 class. That's introductory genetics. I hope we are keeping safe in our various locations. Before we went on break, we started to describe the chromosome structure, and based on that, I will continue from there. Chromosome structure. Now, looking at the typical chromosome, you have its position of gene, then you have the sister chromatids. The sister chromatids are joined together by means of centromere. When it is unduplicated, you have it as single, and when it is duplicated, you see that it is double. All those lines that you are seeing, they are the positions of genes on the chromosome. As we know that chromosome is very important because they carries the genetic materials. That's where the gene, that's what houses the genes. Now, at the mitotic metaphase, chromosome consists of two symmetrical structures that is called chromatids, as we have seen in the last diagram. And each of the chromatids contain a single DNA molecule. Both the chromatids are attached to each other by centromere, which will become separated at the beginning of anaphase during mitotic division. The typical chromosome consists of a chromomere, centromere, and then telomere. So what is the chromomere? They are bead-like structures. That is, they are the accumulation of chromatin materials, which are sometimes visible at the interface or interface. Then, why are they called chromomeres? They are regions where you have a tightly folded DNA, and they become, especially you see them, they become prominent in a polytene chromosome. Polytene chromosomes, they are giant chromosomes. They are not just ordinary chromosomes, they are big chromosomes. Now let's look at centromere. The centromere in a chromosome contains specific DNA sequences with special protein that is bound to them. So also in the centromere, you see you have DNA and some special proteins. So centromeres are those condensed regions. What I mean by condensed, that is, they are packed together within the chromosome. They are responsible for segregation of replicated chromosome. What I mean by segregation, that is separation of chromosome when they have duplicated or when they have replicated during mitosis and meiosis. So centromere is very important during the separation. And let's look at the telomere also. It is the chromosomal ends which prevents other chromosomal segments to be fused with it. They are the region of DNA at the end of a new linear eukaryotic chromosomes. They are also required for replication and stability of a chromosome. So telomere, they are the tail end of each chromosome. Then let's look at chromatin. As I've explained earlier, I said chromatin and chromosome. The difference between them is just that chromatin, they are tightly packed together, while we have the chromosome being loosely, so that you can chromatin, they are loosely arranged, while on chromosome they are tightly packed together so that you can see them at the metaphase. But in chromatin, they are loosely they are not compact, they are not condensed, and that is why you see the chromomere when they are tightly together. You see the structure of chromomere at the interface regions where those DNA materials, they tightly folded. Now, what do we have in the chromatin also? We have the DNA, so you know that's deoxyribonucleic acid. We have the RNA, we have the protein. The protein of chromatin, it is of two types. Like I told us, it can be histones and non-histones. Histones are responsible for the packaging of this chromosome so that it can give us that structure at the metaphase. While non-histones, they are responsible for replication and as well as gene expression. Now let's look at what DNA. It is an important chemical constituent of chromatin because it plays a central role of controlling heredity, and then it is the major genetic material. The genome of an organism can be described by the number of chromosomes it contains or by the amount of DNA in the haploid cells. Don't forget, I explained what I mean by haploid and diploid to all. So you have them, you have a genome being expressed as the amount of DNA per the haploid cell. Now, this is the structure of DNA and RNA. Look at it closely. Don't forget, I told us that we have cytosine, guanine, adenine, and uracil. Those are the nitrogenous bases. Now, you can see them, cytosine, guanine, then you see this color. So that will be telling you that is for that of uracil. Look at the blue for cytosine. Then you have the green, guanine, then the one that looks like yellow for your adenine. These are the position of the nucleotide bases. Now, the difference you can see in RNA and DNA, as I've said, the RNA is just a single strand, as you can see it, and you see the presence of uracil. That's why you have this orange color. That's uracil. But coming to DNA, which is your deoxyribonucleic acid, they are two strands. I call them double-stranded. So you see that you do not see the presence of uracil again. You have it as thymine. So in DNA, your uracil will be replaced by thymine. So you still have your presence of adenine, guanine, and then cytosine. Don't forget all the rules of base pairing. I told us that the thymine will bind to adenine. You can see it with your yellow and red. So adenine will bind with thymine. Then whereas you have your cytosine binding with guanine. So that's the structure of DNA. Then not only at nitrogenous bases do we have in the DNA, we also have what we call the sugar base. We have the sugar base. I told us it can be ribose or it can be deoxyribose. Then we still have the phosphate groups. So those are the three things that you see in your DNA. So you have your nitrogenous bases, you have your phosphate group, and you have your sugar. That's the arrangement. So when they are not duplicated, they will just be one single strand. And when after the duplication, you see them as being double-stranded. So when they want to replicate, that is when they want to duplicate, what will happen here is that they unwind. As you can see, they will unwind So as they are unwinding, they will be what? They will be pairing, following the rules of base pairing. Now, let's look at chromosome number. The chromosome number, you have 2 copies of each present in somatic cell. I explained to us that somatic cells, they are the body cells, and then you can also call them autosomes. The number of unique chromosomes in each cell is called haploid number. So haploid is the number of unique chromosomes, and the total number of chromosomes, which is 2n, is called diploid number. So now the haploid set of chromosome is known as genome. Don't forget I explained the genome the other time. So the haploid set of a chromosomes, it is known as the genome. So the genome of an organism is the haploid set of chromosome of that particular organism. So the number of chromosome in each somatic cell is the same for all members of species. Like in human being now, it is 46. So it doesn't mean that it's going to change in other, it doesn't mean that it's going to change in maybe another person unless mutation occur. So it is the same. Then we have the organism that has the lowest chromosome number is the nematode. You have it in nematode, it's just having 2 chromosomes. Whereas in human being, we have 46. We have in wheat also, we have like 46. So we have in plant, we have in males, like Drosophila, we have 8. So each of them, each chromosome number, it varies from one organism to another, and it doesn't change within the same members of species. Let's look at how human chromosome looks like. This is the arrangement of human chromosomes. You have it, if you start counting, you have it as 1, 2, 3. Here, they occur as pair, and it is 23 pair. So the only difference that you see here is that, now look at this one, it is the X. Then you have it, then you have your Y. This is how, then you can see that you have some regions that are tightly packed, and you have some regions that are light. You have some regions that are dark stain, and you have some regions that are what? That are light. So the regions that are dark, are you getting it? They are the regions that are condensed. You have condensed DNA there. In those regions, we have a non-coding. They are non-coding regions. In human genome, we have about 3 times 10⁹ base pair. What do I mean by that base pair? Is that your adenine to thymine, then cytosine to guanine. So you have it 3 times 10⁹ base pair of DNA. Now, even the smallest of the chromosome in human being is several times larger than the entire yeast genome. And you have the extended length of the DNA, that is what makes up the human genome. That DNA is about 1 meter long. So how will it now fit into that small space of the nucleus? That's the work of compression, so which is being done by histones. The human genome is distributed among the 24 chromosomes. Now all the genes, all the DNAs that you see, all of them will be distributed across all the 24 chromosomes. Don't forget, you're going to have the 22 autosomes and then 2 sex chromosomes, XX in female and XY in male. The rest are known as, so the 22, the remainings are called autosomes. Now, the haploid human genome contains about 23,000 protein coding genes. the rest, they are non-coding. So that is to tell you that it's not all the information on the DNA that will what? That will lead to, that will code for a particular gene. It's not all of them. So we have those that will code for a gene. We call them exon. And those that will not code for a gene, call them introns. Let's look at types of human chromosomes. This is the diagram of human chromosome that is viewed at the metaphase by a powerful microscope. This is how you're going to see it. Then you can see that they are not of the same size and they are not of the same structure, and that's what takes us to the type. You have those that are metacentric. Metacentric, they are having their chromosome at the center, giving us a V shape. Then you have those that are submetacentric, whereas you have one short arm and one long arm. So you have them like J, you can have them as J shape or L shape. Then we still have some that are acrocentric. Those ones, they are having the centromere close to the end of a chromosome. So virtually one arm is very long than the other one. And also we have for telocentric, whereby the centromere is at the tail end. These are the 4 types of human chromosome that you can observe at the metaphase. I told us that you can only see a chromosome very visible at the metaphase. Now, what do we call karyotype? They are the numerical description, their number and size in the normal diploid cell. You see how this diagram looks like now. Before they can be arranged, so when they want to arrange this one, they will be picking those ones that are very close to each other, be arranging them. Look at this one and this one, they are close together. So you're going to pick, you're going to cut them, then you can also measure them so as to know that they are of the same size. Then placing them together based on their structure, that's what we call karyotype. So you are going to look for their pair. If you count this one, they will be close to 46. So you will be pairing them based on the similar structures that they have. That's what we call the karyotype. Now, what is the essence of this karyotype? Is because it's very important to correlate changes, so that in case you see maybe like 47 chromosomes or 46 or 48, you know that something is wrong. So that is when you can detect changes. So karyotype help us to detect changes that occur. So the phenotype of individual. So you will have noticed that this is not normal. Are you getting it now? So it will enable us to trace what is actually wrong. So on a good day, women are supposed to have 46. Anything other or lesser than that, know that mutation has occurred, something has occurred. So they have done this one, the human karyotype, that one you saw. So they pair them together based on their structures, so that we can have 22 autosomes. And when it comes to the X and Y, you see that they are having different structures. So you count them. Then, now let's look at variation in chromosome structure and number. Like I told us the other time that anything can happen. So let's look at what if there is changes in this chromosome structure or chromosome number. And the first thing that can cause changes is mutation. So we have mutation at the level of a chromosome. So we have members of the same species have the same numbers and types of chromosome, with only the exception of sex chromosome, which in male you have Y and then in female you have only X. So when mutation occurs at the chromosomal level, it can cause phenotypic changes. And an example of chromosomal mutation we have in the aneuploidy. Aneuploidy is a type of chromosomal mutation which has substantial changes in the chromosome structure that are large enough to be visible. You can only detect this condition by karyotyping the chromosome number, the chromosome. So we have an example of this aneuploidy, which is trisomy 21. What do you mean by trisomy 21? An individual will be having 3 chromosomes instead of 2. So at the 21, look at what I'm saying here. You see that this thing is number 1, 2, 3, 4, 5, 6, 7. Now coming to this 21, instead of having 2 here, you're going to have 3. So this situation is called trisomy 21. And then those individuals with such, we have what we call a Down syndrome. So his or her karyotype will be written, it will be 47. 47 because of the extra one that you have at 21. So, and what causes most is non-disjunction. You have non-disjunction of this chromosome at anaphase. So that means that the 2 copies move into segregate. So they didn't separate. The 2 of them just move. So that's what we're going to have at the telomere. Then we still have another condition, which is polyploidy. If the mutation involves an entire set of chromosomes, so in that case you are going to have polyploidy. This condition is not viable in the human being. So it's not, in fact, before the baby cannot survive the condition of polyploidy. But it do happen in plants. So plants can tolerate polyploidy, and in fact, human beings exploit this. They exploit this condition so as to make, most of the time, to make their fruits big. So you have the case in the seedless watermelon. Instead of it producing seed, so they will make it to work to their fruits. They will direct all the effort to the fruit. So in those cases, you don't have them producing seed. That's the variation for, we have some. We also have another one that it will be reduction. So you have those ones that will be reduction, and we have those ones that will be addition. It is in the case of addition that we just talked about. That is for the aneuploidy. Then let's look at mutation also in chromosome structure. The structure can also change. Then these are the types of situation whereby it can cause changes in the chromosome structure. We have deletions. In this case, you have loss of a chromosomal segment. A just portion of the chromosome will just be deleted. So in that case, it might have phenotypical differences, and sometimes if it is not a gene, it's not a region that is very necessary, it might not have any implication on such human being. Then we also have duplication, that is repetition of a chromosome mass segment. So compared to the normal parent, so you are having double of what you have in the parent. Then we also have an inversion. That is a change in direction. Maybe it's supposed to read A, G, C, T. Are you getting it? It's now reading A, G, T, C, A. Those ones, they are called inversions. Then we still have translocation. A chromosomal translocation occurs when a segment of one chromosome becomes attached to another. So a segment will be removed completely from a chromosome and it will be joined to another one. So in such case, we have a translocation. So these are the possible situation that can happen that will change the chromosome structure. You have your deletion, duplication, inversion, and you have translocation. But in numbers, it is either subtraction or you have it in the addition. So I want you to go over the material that is with you. You can check it again and again. So far so good. I have established what chromosome looks like, and can check the chromosome size. I said the sizes also ranges. The chromosome sizes also ranges from one organism to another. Then shape. The shape of the organism will be determined by the position of the centromere. Then your genome, which is the haploid set of a chromosome in an organism. So with that, we've come to the end of the class. Thank you and keep safe.
Finish him. Kill the guy now. Grab the gun. Hit him.
What? What is this?
Good day, sir.
You are watching Jackie Pan in my house.
No, it's King Kong, sir.
King Kong. You've never heard of ACTV before in your life? ACTV is very educative, informative, and there are football sport there. There is entertainment. There are a lot of things you can even attend lectures here. All you know is watch all those nonsense artists and do nonsense, or you watch nonsense on TV. Last night you almost came in bed with your fist.
It's not me, sir.
And you wake up every morning, the bed will be so rough, like you've been fighting kung fu in your dream. That's the reason why you have F's in your courses, like especially made for you.
I have it last semester.
We eat by pounds on you. Very stupid though. Yeah, change that section to ACTV right now. And oh God, the baby wants to have the first born. Stupid boy.